Hello, everyone. It gives me great pleasure to present my poster at the 51st Annual Meeting of the American Pancreas Association. Over the next couple of minutes, I will show ALCAM as a novel CTC marker for PDAC. EpCAM and cytokeratin are the current gold standards. There are a lot of problems when it comes to EpCAM as being a CTC marker, and there's a huge variability that's seen in the field. The need of the hour, therefore, is to globally characterize CTC surface markers. We also do need tumor cell markers that reflect the biology of the cancer. In order to do this, we basically isolated CTCs in an unbiased, marker-independent method from a PDAC cancer patient with METs. We identified multiple ethnic races and screened them for EpCAM negative cytokeratin positive cells. And then we did a flow cytometric and a mass spectrometric screen to identify candidates that we validated in KPC GEM and metastasis models. We also validated them in PDAC patients. We identified ALCAM as a type 1 transmembrane glycoprotein as a potential PDAC CTC marker. This is a protein that is normally seen in intracellular junctions or also in activated T cells and monocytes. It is a cancer stem cell marker. The differential cleavage of these two isoforms determines an overabundance of the cytoplasmic part, which has been known to increase metastasis in colon cancer. We wanted to assess its expression in the KPC mouse model. So what we did see was overexpression of cytoplasmic ALCAM in PDAC pancreas. Further, the localization of protein changes from membrane to cytoplasm in aggressive PDAC. We wanted to see its conservation in METs. We looked at liver, lung, and splenic METs. And overexpression of cyto-ALCAM was also observed in sites of distant metastasis, indicative of it being a bona fide metastasis marker. We wanted to eliminate the source of other cells contributing to this, so we did a simple experiment. We took the KPC GEM mice, isolated CTCs from the ascites and blood, and injected them into black 6 mice. We looked at the tumors that resulted, and we do see that the source of cytoplasmic ALCAM are the CTCs isolated from the ascites or blood. Localization of the ALCAM in the tumor is also cytoplasmic, indicative of faithful preservation of expression. The proof of the pudding was to see if we can actually validate these markers in PDAC patient samples. So here are 2-3 patients where we do see cyto-ALCAM being expressed in single cells or even in clusters. Expression of the marker does not change in a patient who has had chemo, indicative of robustness of the marker, even with treatment. The working hypothesis is that the differential cleavage of these two isoforms by ADAM17 allows the accumulation of cytoplasmic ALCAM in the cancer cell, which mediates its extravasation from the blood vessel and entry into a distant metastatic site. The future directions are basically investigating the nature of cyto-ALCAM in patients with respect to progression and therapy, investigation of signaling pathways that are associated with this protein, and further validation of cyto-ALCAM and other GI cancers due to its stem cell origin. Thank you.